to this fourth Zurich Immuno-Oncology Symposium. I think we have an exciting program. You know, we have, the last 3 years, we have preliminary reported on advances in the treatment. We will continue to do that, but we also thought it would be important to touch practical aspects during treatment, patient aspects in this symposium today. Before I start, I want to say some thanks. I want to thank the sponsors, and they're listed up here. So we had very generous sponsors to support this meeting. I want to thank, just right now, the faculty who has taken time to prepare and come to Zurich. And then I want to thank and give a hand to Kathleen, who has done everything in communication and logistics to make it possible. Thank you, Kathleen. As usually, we start with a keynote address. We're very lucky here to have Burkhard Becher from the Institute of Experimental Immunology in Zurich. He'll talk about single-cell screening in precision medicine. Burkhard, please.
The right button. So thank you so much for having me, and thanks for the introduction. And it's a pleasure to be here. And it was a relatively short trip for me, which is wonderful. So I'm one of the faculty lucky to not have had to travel much longer than half an hour or something like this. And I would like to talk today about basically a method, something that is perhaps less familiar to those of you who treat patients, something that you may be less familiar with, but it is something that we think is rather important and will be more important in the future for decision making, but also to understand a couple of fundamentals. So I'll talk about two things quickly, and I try to make it somewhat clear where I'm coming from with the example of your early morning breakfast. When you're deciding to have a smoothie for breakfast, your decision is, do I have a smoothie or do I have a fruit salad? And the reason I bring this up is because there was a revolution basically in biomarker discoveries and patient stratification and understanding the pathophysiology of diseases, including cancer, about, I'd say about 20 years ago, with the dawn of the so-called omics era. Omics basically means you're looking at not just one single molecule that you're interested in, but you interrogate a whole tissue or liquid biopsy or blood, for instance, for all kinds of different parameters, including the entire transcriptome. You can actually sequence every single RNA molecule there and what is expressed, and they give you an idea about the state of the affairs in this particular situation. The disadvantage is, and that's why I bring this example of the breakfast, is the fact that you're looking at a bulk result. If you're looking at peripheral blood, it turns out that all of us have a different composition in our peripheral blood to begin with. So if I interrogate the peripheral blood as one entity and then look at the transcriptome or the proteome or the metabolome or what have you of your peripheral blood cells, it turns out I get basically a bulk result that is akin to what you see here, the milkshake, rather than the single cell entity. Now, if you think about that maybe your disease is caused by a very rare subset of cells, something that is not very abundant. It's very, very rare, like here in this case, only these two cells, which are different, and you would like to know what these cells look like, what they are, and how they behave. You do need single-cell data, because the vast majority of the things you're looking at are bananas, but you do want to have these two strawberries. Now, it's a question of sensitivity of your tool. If your tool, and in this case it's your eyes, your mouth, the tongue, your ability to smell, if you have in your milkshake 1 or 2 strawberries, it really doesn't make a big difference. Even if you have a strawberry or a raspberry, it'll give you the exact same result in taste, in look, in flavor, unless, of course, you are particularly good at identifying single fruits in your milkshake, but I doubt that you are. Now, having said that, with the premise, and without going into the technology aspect here, we like to look at individual cells, and we'd like to look at a lot of different things on one single cell. And it turns out that we can potentially discover a couple of items. And so one of the fundamental premises, things that we do, is we compare different groups, and this what I'm showing you here is all patient-based. So everything is about, let's say we have 3 different groups of patients. And in these patients, we then harvest single cells from these patients. Usually, you do this from the blood, but you can also use cerebral spinal fluid if you want another liquid biopsy, but sometimes even tissue. And in the second part, I'll talk quickly about when we look at human brain, what that looks like on the single cell level, and give you an atlas of the human brain in the context of cancer. Now, we then barcode these single cells, so each individual patient gets an individual barcode, so we can identify the patient later on after we've done the analysis. Then we use what we call single-cell cytometry, in which we can interrogate usually proteins, up to 100 proteins, for instance, or we can look at RNA transcripts, depending on the depth and how much money you're willing to spend. So you can find out a lot of different things. And then the key here is to recognize our inability to recognize patterns. So our own stupidity is the reason why the next step is then performed with the help of a computer. I'll get a little bit into this in a second. Actually right now. And the reason why we need computers is because of our inability to think more than 3 dimensions. So if you have one parameter that you're looking at, one molecule, one protein, it can be positive or negative. It is present in the cell, in this one single cell, or it's negative. So 1 dimension will give you 2 levels, positive or negative. 2 dimensions, and geometrically the equivalent is an area, like in this case here, this 2-dimensional area, gives you 4 levels. 3 dimensions, already 8 levels. So with 3 parameters that you're looking at one single cell, you have 8 levels of information if it were just plus, yes and no, or plus or minus. 4 dimensions, I guess you cannot imagine 4 dimensions. Very well. So here, mathematically, the way it's done is you just put 2 cubes next to one another and you fake 4 dimensions. But when we look at 40 and more dimensions, it becomes not imaginable anymore. We have like more than 1 trillion levels, which is way beyond our capacity to think and compute. In practical terms, for those of you, and specifically some of the clinicians may remember in the old days when you had to work in the lab for a short period of time, here's a good example of flow cytometry. In the first case here, we're looking at one plot that has 2 parameters, on the x and on the y-axis. A molecule that is labeled so that it can be revealed on the x-axis, and another molecule that is revealed on the y-axis. Now, this can be analyzed by a monkey. That's relatively easy. You and I, all of us can do this. I used to do this. I still do this. It works. 3 parameters, easy. You just add another one. You can even make it 3-dimensional. With 9 parameters, as you can see here, to do it right, it already becomes quite complicated. But if you're looking at 30 and more parameters, this is beyond the human capacity to compute data. So what we do is we use different tools, and I will not go into details here, but I can tell you that this is definitely a revolution in my area, that computer-aided analysis of our data can reveal things that we could have never seen before, because we had a very focused, biased vision on what we want to see, what we expect to see, what we were looking for. But now we're not looking for anything. So this is basically the next set of this is what you call pattern recognition. And I have a wonderful example. Those of you who have a smartphone, I'm sure everyone here has a smartphone and the capacity to take a picture, a foto. So when you go and take a picture with your smartphone, what ultimately happens is you will dump all of this on your computer, and most of the computer software will ask you, would you like us to split your pictures according to who is on the pictures? So you'll have pictures of your mom, of your sister, of your brother, maybe of your children. I have actually pictures of my own daughter, and the software has learned, because it's a learning software, to recognize my daughter from the age of 2 to now the age of 21, and it categorizes it with almost no mistake. Sometimes it mixes in my wife, occasionally my son, once or twice a dog, but usually it makes very few mistakes. So, and this is what we then apply to our data. So, and I give you just now 2 sets of experiments that we've done. here in Zurich together with colleagues at the clinics. So the first one was we were thinking, as you know, and I don't have to explain this to this audience, that patients with melanoma who undergo immunotherapy, and in this case we were looking at blockade, neutralization of the PD-1 molecule, have a relatively high chance of doing rather well under the therapy and go to the stage that we call progression-free survival. Now there is, however, it's not 100% of the patients, it's actually not even 50% of the patients. It's right now, I think the areas, we can discuss it, maybe about 40%. What about the other 60%? They have the side effects, because this isn't sugar water, anti-PD-1 has side effects, so these people have the side effects, but no real clinical benefit. So we thought, would it be possible to select patients prior to therapy and only treat the ones who are most likely to benefit from anti-PD-1 therapy, and the patients who are not likely to benefit, maybe offer them an alternative therapy, which are available and they are being tested right now. So we thought that the patient stratification does not necessarily mean, well, there are just upsides to it. It doesn't mean we don't treat that patient, but the patient will not be burdened with the side effects and will not get a therapy that she doesn't benefit from. So that was the idea, and the method that we used here is we used a machine called the mass cytometer. Sounds really complicated, is very expensive indeed, and not everyone in the clinic can do it, but ultimately we want to translate that into the clinical routine, that every patient can be measured prior to the therapy by more simple means. Anyhow, so we use the pattern recognition software, a pattern recognition algorithm that teaches itself. It's what you call a neuronal network-based computer algorithm. And you look at the data here, and of course this is not very meaningful, but what you may be able to see here, and I basically boiled it down to this, is that patients who respond prior to being treated have a profile in a population of cells, of white blood cells, of monocytes, that we could then validate later on in patients. So we could find out, yes, these patients before therapy have a higher number of these monocytes and are much more likely to respond to immunotherapy. Now, what we need to do now, and this is actually the other challenge, is to translate that into the clinics, and that means we need to work with clinicians and convince them that we want these data, we need these values for us to validate our own findings, ideally not just in Zurich, but in the whole world. So any of you interested who treat patients who are not in Zurich, I'd be happy to work with you and look at your data that you have in the routine just for us to analyze and look at later on. And the other example, and basically I'm more or less finished with this then, is brain cancer. And this is data that is not yet published. And I'm very excited by this because I told you what we usually do in the context of treating patients and looking at patients is we look at liquid biopsies, peripheral blood mononuclear cells usually. But we had the opportunity in collaboration with neurosurgery and neurology here in Zurich to have access to brain cancer samples. And so there is different ways. So brain cancer is a big area. The most of it's often called glioma, which can be then astrocytomas and glioblastoma in different grades, as you see here. And oftentimes there are, can be separated based on the mutation of IDH, which again, I think you'll hear about it much more later from the specialists, which I am not. But another kind of brain cancer, the brain is a wonderful place for metastasis to grow, apparently. So it's something that a lot of different cancers do actually spread into the central nervous system. And there are a couple of simple questions we wanted to ask because I think it's fundamentally important to understand a few things about this. First of all, so what shapes the so-called tumor microenvironment, which is not the tumor itself, but it's the fibroblasts, it's the immune cells that infiltrate, the immune cells which are present. Is there a difference between a metastasis that moves into the brain, if it comes from the breast, for instance, to a metastasis that moves into the bone? Does the brain shape the microenvironment, or does the tumor itself decide what it looks like? So, in other words, whether the tumor microenvironment in the brain is shaped by the brain tissue or the nature of the disease-causing cancer. And there's another thing that you need to know, let's call it a bit of a revolution in our understanding of these cells that we call tumor-associated macrophage. It's a really hot topic. A lot of people are talking about this, but it's a very diffusely described entity. The brain, as you may know, has macrophages that live there at all times, and their normal function is probably to help us Actually, there's the theory that our brain macrophages are the reason why we have dreams at night. So that's one of the things, because they nibble on our synapses during the night, and that actually causes us to think of weird things. Or actually, a dream is not thinking, but you have dreams. And that's basically, there's some good data for that. But the point is, these brain macrophages are called microglia, are not derived from monocytes, like many that most of us used to believe, but basically, most tissue macrophages are embryonic derived. So during embryogenesis, our tissues have these macrophages. They live there, and they're part of the tissue and have specific tissue function. In inflammation, or in cancer, or disturbed homeostasis, you have influx of monocytic cells, monocytes from the blood go into the tissue, and then they behave like macrophages, they look like macrophages, and actually, even for the best of the pathologists in this room, they're virtually indistinguishable from the macrophages that live there. So you have already at least 2 types of tumor-associated macrophages, and we thought maybe we can distinguish them. So the experimental workflow here is that we had tumors from 37 patients with cancer, mixed between glioblastoma, low-grade gliomas, but also epilepsy patients and patients with metastasis. And we then did the same experiment, this time looking at the cells that we got from the brains. So we take the brain tissue, dissect it, make a single-cell suspension, and analyze it, and interrogate it, in this case, 74 parameters. And the first thing we did is what you call a principal component analysis. It immediately said, by just looking at the raw data, the computer was not told what to do, but said, do you see something that's different here? And the answer is yes. The glioma patients all hang on one side, whereas the metastases are pretty much all on the other side. And that is probably the cleanest PCA that I've seen. So it's relatively clear there's something fundamentally different between a brain metastasis and a primary brain cancer. This is what the data look like. Again, I will not go into detail here. We find a lot of different cells. We are, as immunologists, obviously a little bit biased because we like to look at the cells that we think matter the most, which are the immune cells. And in the context of this symposium, I think it's not entirely inappropriate. Anyway, so one of the things that you see right away, the big difference between patients with brain metastasis and patients with primary brain cancers is right here, and that is the number of T cells that invade. So the more the tumor comes from the outside, we think the more it is recognized and brings along a whole baggage of immune cells that try to get rid of it. I'm not saying that this works very well. Obviously, this is not a good thing to have brain metastasis, but the presence of T cells in these metastases gives hope for the possibility of treatment. Now, this is basically quickly the data. But the other thing that I told you, we're able to distinguish embryonic-derived macrophages, the microglia, very clearly from the macrophages that are coming in from the immune compartment, from the blood. But first, let's look here quickly only at the microglia compartment, the brain-resident cells. How do brain-resident macrophages, these microglia, behave if they encounter a glioma or encounter a metastasis? And the answer here is relatively clear. In low-grade glioma, it looks basically very similar to what we see in epilepsy cases. Now, I'm not saying epilepsy is a perfectly normal control, but it is the best we can do in the lab if somebody is undergoing surgery for epilepsy, because it is unlikely that any of you are willing to donate brain tissue for our controls here. So we think that the epilepsy and low-grade glioma, the fact that they look so similar, basically tells you that the microglia do not really recognize that anything is different here. The moment that, as you can see here, there is a bit of an evolution in the behavior of the microglia compartment. With higher grade gliomas, these cells look very, very different. And then when you have a metastasis, this is probably the most dramatic change here. The cells have changed dramatically. Now, it becomes even more clear when you look specifically at monocytes. So monocytes are basically just these few cells that start here. When they enter the tissue, they change their phenotype and become these macrophage-like cells. And you can see again, there are very few monocytes present or monocyte-derived cells present in low-grade glioma or in epilepsy, and most of the macrophages are in fact sorry, I'm just stuck here. Most of the macrophages are in fact these microglia cells. And as you can see here, in higher grade glioma, it becomes more and more cells are moving in. This is in astrocytoma, so in glioblastoma, you have these cells moving in and behave very differently, and these are not microglia. These are microglia. These are, in fact, these macrophages, monocyte-derived macrophages, and that we can clearly distinguish them. But it becomes most dramatic in melanoma, and in this case here of lung cancer, where you see this massive influx of immune cells into the brain and the transformation of these cells into these strange-looking tumor-associated macrophages. Again, so we made sure that we can phenotype the cells. There are actually markers and reasons why they are differently positioned on this map that I show you here. This is a single cell map. Each little dot is a single cell, and the fact that they are at a different position means that they look different. And what is different about them, we can actually extract this information and test it in large databases as to whether this is whether they play a role in disease progression or not. And in fact, the data match very much what we see with the genomics data from the TCGA database. Now, last little bit of information, and a big surprise to us. What we found the most surprising is the presence of these cells that you will hear more about today, which are so-called regulatory T cells. So we have the assumption that regulatory T cells, their main driving, the main reason for being is to limit immune responses, to block immune responses. So in the context of cancer immunotherapy, regulatory T cells are a bad thing. However, we need to also understand that regulatory T cells are heavily attracted to inflammation. So the presence of Tregs, regulatory T cells, is perhaps a bad thing in the context of cancer immunotherapy, but also a sign of something going on, of inflammation. And so we do believe that these data and the presence of the high presence of regulatory T cells and metastasis may very well be the reason why the tumor can progress. But if you were to target this particular aspect here, you have probably a much better chance, like for instance, there are antibodies to regulatory T cells that deplete regulatory T cells, such as ipilimumab or CTLA-4, that might be a good thing to use in a brain metastasis, but it would not really give you much benefit in glioblastoma because there are no regulatory T cells. There are very few T cells to begin with. Anyway, I would like to just basically put it together here so that we have maybe time for maybe a question or maybe not. It's just simply that what I wanted to showcase here is that this method that I'm showing here, the interrogation of single cells, may they be from the tumor or can they be from the patient's peripheral blood, does, in this case here, just to talk about the last bit, in the context of brain tumor, will actually tell you about the diversity, the composition, and to some extent the behavior of the brain tumor microenvironment. The monocyte-derived cells do accumulate, particularly in the more severe glioma environment and glioblastoma environment, and dominate among the myeloid cells and metastatic lesions, whereas in the lower grade tumor, in the lower grade glioma, you see basically microglia are actually the main population that we find. And the composition of tissue-resident lymphocytes that people argue about, and you can't easily do this just by looking at tissue slides that are available to you. You really need to go much deeper in detail in order to understand, and maybe, at least this is very preclinical work here, but maybe we can go and make some informed decisions about how to treat a patient, not necessarily after we have harvested a bit of brain, but after we know what is the tumor entity, what's the composition there, how to best treat and how to best alter the immune response. Now, the last thing I want to share with you is basically my team that is doing all of this work, because I have done nothing here. It's actually really, really nice to not have to work anymore, so it's good. So That's all I do sometimes. No, but in fact, the people work pretty hard, and it's an excellent team right now. I probably have the best team I've ever had. And I have a wonderful set of collaborators that you can see on the left side. And in Zurich, in the first part I talked about, the melanoma work is together with the Clinic of Dermatology, and the glioblastoma work is together with the neurology group of Michael Weller, and surgery. The neurosurgeon here is actually Maria Neidert, who provides us the tissue and who sits down with us and we discuss data. And I've been speaking for 21 minutes, and I thank you for your attention.
Thank you, Burkhard. Even if you do not work anymore, I think you make nice talks. And it was very physician-friendly talk in explanation the complexity of research. We will have time for 1 or 2 questions. Michael?
This is nice talk. It's completely off the topic, but is there really any data on my synapses, my macrophages, and my dreaming? That's really interesting.
Yeah, I didn't know that either. So you think that, I mean, in the old days we used to think that microglia, the brain-resident phagocytes, their main function is immune-mediated, that they are involved in immune defense. I think the evidence for this is basically fading away into oblivion. There isn't much left there. But why are we having these microglia? And the only thing that's left at this point that seems to be important is synaptic pruning. And apparently synaptic pruning happens mostly, at least in mammals, during the time when you sleep. And this is when you have these REM phases, and there is evidence that the synaptic pruning leads to you thinking that you can fly or something.
Good, thank you, Burkhard, and I hand over to